Hello friend. A video making competition was organized by SCERT Education Department of Maharashtra for the teachers. In this, I got first prize at divisional level Mumbai. And second prize at state level.
राज्यस्तरावर विज्ञान विषयाकरिता ठाकूर विज्ञान व वाणिज्य महाविद्यालय कांदिवली पूर्व मुंबईमधून श्री प्रवीण पांडुरंग भोसले यांना पुरस्कार प्रदान करण्यात येत आहे.
The award was given in the form of cash prize, certificate, trophy and medal. So everybody were curious about the video. So here is the video.
Double fertilization. The chapter is reproduction in lower and higher plants. In double fertilization, fertilization takes place twice. Here, one male gamete fuse with female gamete and second male gamete fuse with secondary nucleus. So twice there is fertilization, and that's why it is called as double fertilization. It's a important characteristic of flowering plants, angiospermic plants. This process was discovered by S.G. Navasin in the liliaceous plant like lilium and fritillaria. After pollination, pollination means transfer of pollen grain from anther to the stigma. So after pollination, pollen grain reach the surface of the stigma. It germinates and form pollen tube. This pollen tube penetrates the stigma, style, ovary chamber and then enters ovule. The growth of pollen tube is guided by the chemicals secreted by the synergid. Synergids are here in egg apparatus. Entry of pollen tube is by three different modes. The common one is the pollen tube enters through micropyle. This is termed as porogamy. So when pollen tube enters through micropyle, it is porogamy and it is a common mode of entry. In some cases, the pollen tube enters through chalaza. It is called chalazogamy. And in some plants, the pollen tube enters through integument. It is called as mesogamy. So when pollen tube enters through micropyle, it is porogamy. It is common type. In some, it enters through chalaza. It is chalazogamy. In some plants, it enters through integument. It is called as mesogamy. Finally, pollen tube penetrates embryo sac of ovule through its micropylar end. The pollen tube carrying male gametes penetrates in one of the synergid. Watery content of synergid are absorbed by pollen tube, which then ruptures and release the content including the two non-motile male gametes. As non-motile male gametes are carried through hollow pollen tube, it is known as sphenogamy. So non-motile male gametes are carried through hollow pollen tube. It is called as sphenogamy. Fusion of one male gamete with female gamete is called as syngamy, and fusion of second male gamete with secondary nucleus is called as triple fusion. Syngamy and triple fusion are two events of sexual reproduction in angiospermic flowering plants. Syngamy results into diploid zygote because male gamete is haploid, female gamete is haploid. So after fusion, diploid zygote is formed, whereas triple fusion results into PEN, primary endosperm nucleus. Here male gamete is haploid, secondary nucleus is diploid. So pen which is formed is triploid, and this pen, primary endosperm nucleus, further develops into triploid endosperm. Zygote develops into embryo. Significance: it's a unique feature of angiosperm, ensures that the parent plant invests a seed with a food store only if the egg is fertilized. The diploid zygote develops into embryo, which consequently develops into a new plant. The triploid pen develops into nutritive endosperm tissue. It restores the diploid condition by fusion of haploid male gamete with haploid female gamete, that is through syngamy. It also helps to avoid polyembryony. So this was all about double fertilization.